Hey, this is Dr. Cummings from PLNU, and we're going to start a short series of 3 parts, 3 video lectures on antibiotic resistance. If you've not yet studied how antibiotics themselves function, you're going to want to go back and watch that 3-part series. It's the episodes immediately preceding this one, because it's really important that you understand those antibiotic targets, the way that an antibiotic works, because only then can we really understand how the bacteria protect themselves from those antibiotics. So I'm going to remind you of those antibiotic targets briefly, and then I want to differentiate those strains of bacteria that are non-susceptible to an antibiotic versus those that are actively resistant, those that should otherwise be susceptible but have acquired some sort of resistance. And then we'll talk about how they can acquire that resistance, either by mutation or by horizontal gene transfer. And if, in fact, we'll eventually focus this series on those acquired resistance genes that are shuttled by horizontal gene transfer, because it appears that those are arguably the most important. They seem to be the key behind the explosive spread of antibiotic resistance that we've seen around the world. So that's where we're going to focus our time right now. I used this slide in one of the previous videos to talk about the different mechanisms of antibiotic resistance. If you need to pause the video, or pardon me, mechanisms of antibiotic action, if you need to pause the video here and review these, now would be a good time. If you remember, our most common mechanism was to inhibit cell wall synthesis, right? So peptidoglycan was the target. The process of translation, producing proteins from the information on a messenger RNA, is the next most common. And then we have 3 others in no particular order. We've got the cytoplasmic membrane disruptors, especially the polymyxins like colistin. We have the antifolates. These are the sulfa drugs that knock out a metabolic pathway for producing folic acid. And then we have the DNA and RNA synthesis inhibitors, and in particular you need to know the quinolones and the rifampins. So with that in mind, let's think about how bacteria can protect themselves. First of all, let me first say that, like we talked about with antibiotics, each antibiotic has a spectrum of activity, right? No one antibiotic is able to kill all and any bacteria. And it's not because these other bacteria are necessarily resistant. It's more accurate to say that they're non-susceptible. They have some sort of innate resistance, if you want to say that. And that's either going to happen because of a lack of the antibiotic target. So, for example, the chlamydias have little or no peptidoglycan. Well, if they have little or no peptidoglycan, do we say they're resistant to penicillin? No, we'd probably say they're non-susceptible, because they don't even have the target in the first place. So they're not doing anything actively. Nothing has changed that protects them from the antibiotic, but the target of the antibiotic doesn't even exist. So there's an example. Or in some cases, we see some non-susceptible strains arise because their membrane has real low permeability. And we especially see this among gram-negatives. For example, species within the genus Pseudomonas. Pseudomonas species, SPP is plural for species, are notorious for being able to pretty rapidly adjust the permeability of their LPS layer. So if they find themselves in a relatively toxic environment, they can, in a sense, shut down diffusion across the membrane until the environment is safe again. So in both cases, we wouldn't consider those necessarily to be resistance mechanisms. They're really just sort of a lack of susceptibility in the first place. Now, how does that compare to when bacteria are completely resistant? Well, here's the examples I just gave. Right, you've got a gram-positive here. There's peptidoglycan. In a gram-negative, you've got peptidoglycan, but it's hidden. It's sandwiched between the LPS layer and the cytoplasmic membrane, and so there's some non-susceptibility to natural penicillin. So, for example, natural penicillin doesn't get across the membrane very readily. So it just gives you some ideas about how the basic structure of some bacteria can lead to a degree of non-susceptibility. Now, when we think about acquired resistance, okay, this is active resistance, where for all intents and purposes, the bacteria should be susceptible. The membrane is permeable enough for the antibiotic to get across, and the target is there, right? It should be sensitive to this drug, but the bacteria have acquired some way to resist it. So the first, and this is probably one you've learned in some other biology class, is by chromosomal mutation. So on the far left, you've got a population of bacteria. You hit it with a selective pressure, in this case an antibiotic, and just through random chance, due to mistakes made by DNA polymerase is not perfect and it introduces errors, and every now and then one of those errors, one of those mistaken nucleotides may actually result in a protein that is then less susceptible. So the target is still there, but maybe the target is less susceptible, for example. And we end up with a resistant population because the selective pressure kills all those cells within that population that don't have the mutation, and only the one that survives, in this case the blue one, can repopulate. And so now we've gone from a population that was largely susceptible to one that's completely resistant due to first errors in DNA polymerase followed by binary fission. Now we call this vertical transmission. Vertical transmission of any genetic information is from parent to offspring. So if this is a parent bacterium and this is the F1 generation, the offspring receive the DNA from the parents. This is how you and I got all of our DNA, right? From our parents to us, vertical transmission. Let's see if I can get this to move. My slide doesn't want to move now. There we go. Bacteria can also acquire resistance by horizontal gene transfer. And we talked a little about this already, so I just want to review how horizontal gene transfer works. Instead of parents to offspring, now we've got from one, we'll call it a parent if you will, to another. From one cell to another in the same generation, DNA being transferred. And there are three main mechanisms that we learned about where horizontal gene transfer can take place. The first is transformation. When a bacterial cell dies, it bursts open and its DNA gets chopped up into fragments. Small fragments, roughly less than or equal to 15,000 base pairs, can be taken up by some bacteria. Now, not all bacteria are capable of doing this. The ones that can are called competent bacteria. And those competent bacteria can essentially sample the metagenome, the genome of fragments of DNA surrounding them from all the dead cells. And if something happens to be beneficial to them and they can use it and express the genes on it and it benefits them, then they can find a way to keep it. One way to keep that is to integrate it into their own chromosome by a process called homologous recombination. When homologous recombination takes place, then that DNA that they picked up horizontally can then get transmitted vertically from parent to offspring. So when this cell here goes through binary fission, the new DNA will get transferred to the new daughter cells. And so the new resulting population will all have the new genetic information. So that is transformation. The next is transduction. Transduction, if you remember, is when bacteriophage, viruses that can only infect bacteria, inadvertently, really by accident, through the process of the bacteriophage infecting multiple cells, as the phage leaves one cell, it can potentially infect the next and inject DNA from the previous bacteria. Now, most of the time that's not going to make any difference, but every now and then, the DNA they inject from the previous bacteria will be novel, it'll be useful, and they'll keep it and integrate it. And we see lots of examples of changes to bacterial genomes through bacterial transduction. And then finally, conjugation. Conjugation is arguably the most common, abundant, frequent, important mechanism of the horizontal transmission of antibiotic resistance genes among bacteria. So a plasmid we know is a separate piece of DNA, circular, very small relative to the bacterial chromosome, and distinct from the chromosome. And the plasmid, in many ways, acts independently of the host cell. And so the plasmid has genes encoding the information necessary to conjugate, to reach out to a recipient cell that doesn't have the plasmid, to make a copy of the plasmid and to send it over to the new cell, to the recipient. So then the recipient cell itself also becomes a donor. And any accessory genes that that encode things like antibiotic resistance that are found on the plasmid get transmitted to the recipient cell. And so if this original cell was, let's say, penicillin resistant, the recipient cell will now become penicillin resistant, even though it wasn't before. So bacterial conjugation, acquired resistance by horizontal gene transfer. And so the plan as we go through the rest of this three-part series on antibiotic resistance is to focus on the acquired resistance genes that are typically found. We're going to call these acquired resistance genes, or ARGs, that are typically found on plasmids, because these appear to be the most important mechanism for bacteria becoming resistant to antibiotics. Okay, let's wrap up this lesson. Make sure that all the key takeaway points are clear to you, and we'll move on to the next one. So, some bacteria are non-susceptible to an antibiotic. You could sort of think of it as resistant, but they're not doing anything to actively resist the antibiotic. The antibiotic just has no impact on them. Some bacteria are indeed actively resistant to an antibiotic. That active resistance might come from chromosomal mutation that's then passed along through vertical gene transfer, or that active resistance may come from horizontal gene transfer, HGT, by way of transformation, transduction, or most commonly conjugation by way of plasmids. So when you think horizontal gene transfer, the number one mechanism for moving these ARGs, these antibiotic resistance genes, is going to be through plasmids. So if plasmids are a little gray for you, a little foggy, you're going to want to go back and review all the information on plasmids that you can. So after you've sort of absorbed all this, if you've had to watch it a few times, you're cross-referencing your textbook, go ahead and move on to part 2 of this short series on antibiotic resistance.